Greetings, friends of astrobiology. Welcome to a brand new episode of Ask an Astrobiologist, a show where we celebrate science and celebrate scientists. My name is Sanjoy, and this program is made possible by contributions from the NASA Astrobiology program and the nonprofit Blue Marble Space. Last month, I had the great privilege of speaking with Dr. Colin Goldblatt of the University of Victoria, and we talked about a bunch of different topics going from the early Earth all the way to the exoplanets, using the tools and insights that come from atmospheric sciences. And today, we're going to do the same journey, but looking on much smaller scales at the tool of molecular genetics. And my guest today is going to be Dr. Betül Kaçar of the University of Arizona, which we're very excited to talk to. But first, it's time for your field challenge quiz, the competition formerly known as the background quiz. But right now, my studio is my kitchen, so we have to do something new. So Mike, if you could put up the field site challenge for this month. Many of you got it right. As you can tell, it is Lake Salda in Turkey. It is the Maldives of the Turkey, beautiful white sand. And it's very unique. It's a little bit of alkaline water, pH a little higher than neutral, 8 or 9. And that prevents grazers from floor. And thus you have microbial mats that form using a specific mineral called hydromagnesites. Structures are called stromatolites. And some of these stromatolites are some of the evidence of the oldest lives on Earth. Many exist in the fossil records that are over 2.5, almost 3+ billion years old. So these astrobiology field site locations of interest, like the one in Lake Salda, allows scientists to get an insight of how life existed back then. Our big winner for this competition is at Azul Pinoba. Congratulations for getting it right. You will win the astrobiology graphics novel, but in this time of sheltering from home, I have no means to get them to you. But in any case, please DM us your address and we'll ship them to you whenever we can. But you know that you can download all of these graphic novels online, and Mike will set up the URL for you all to see. So thank you very much for playing this week's field challenge. I want to give another big shout out to our ambassador of the month. This is one of you who does more than the usual to promote our program on the interwebs, on Twitter, on Facebook, on Instagrams, and wherever all you might exist online. Thank you so much. This show is only successful because you share it to your friends and family and to your followers. So this month's Ambassador of the Month is, once again, Burfin Formaldehyde, who tweets as @BurfinDagspace. Thank you so much for your support, Burfin. It's awesome to have you in the show. So I'm excited today to welcome astrobiologist extraordinaire Dr. Betül Kaçar, a professor at the University of Arizona. I think one of the very few, if not the only professor in the United States, if not the world, that has a dual appointment in both cell biology and astronomy, a true mark of an astrobiologist. Betül, thank you so much for being on the program with us today.
Thanks for having me.
Like we enjoy doing in the show is turn back the wheels of time a little bit and get back into the child that was looking into space that you were. And if you could share perhaps an anecdote or a story when you were young that got you excited about science and space.
Funny, because I think many of us astrobiologists have that moment where we look at the space and maybe look at the stars and think, okay, what is out there and are we alone in the universe? I am not one of those kids. I didn't even think that a career that involved space and stars was waiting for me in the future. So I would say I was actually thinking about this because I knew you were going to ask me that moment. And so I was doing some soul searching this morning while washing the dishes. I thought, going back, all the way back, I think one of the clearest moments that I had is related to a movie I watched. And it is not about the movie itself, but the way the movie was portraying the scientist in the movie. So that movie is it. And I'm talking and thinking about that movie a lot, where we have the scientist here that is exploring, not the rocks per se, but mysteries that are embedded in pyramids and then gets transported to another dimension and finds out a completely different civilization that wasn't necessarily originated in another planet, but nevertheless evolved rather differently than our own civilization here on Earth. So I think what is interesting about that movie was that it portrayed a scientist that wasn't necessarily a masculine, sort of dorky, character that I couldn't relate to. I was thinking actually how feminine or even actually non-binary the character was, that was different than those generals in the movie who were dictating him what to do and whatnot. So I think I related to that character, and that character made me think that I can do this because I am also someone that was very curious. I still am. I think it started with that, the love for archaeology, the past, exploration, unknowns, knowing different cultures and civilizations.
Wonderful. You are highlighted recently in an article by UN Women, in which there's a beautiful quote by you, which I want to reiterate to our viewers. A girl with determination can change the world. How is it for you, who grew up in Turkey and then made your way to becoming a professor at the University of Arizona, one of the elite institutions in the country, if not the world? Could you tell us a little bit about your academic path and how that went along?
Well, my academic path is not a very sort of straightforward one. And I think you can relate to this. I think who does in the field of astrobiology. I think it takes a bit, not only outside the box, that there is no box, right? The box is not existing. But my career started, I was actually, my undergrad was in chemistry. And my PhD also was in chemistry. So that's a sort of an unknown fact about me, that I didn't start my journey as a biologist, but I studied the chemistry of biomolecules. And I was fascinated by proteins and how they can change everything. And it is true. It's not just proteins alone, molecules. I mean, look at why are you at home right now? Why am I here? Because a molecule is threatening our own civilization here on Earth. They are so impactful. And so to understand that, and I remember the moment, the first time I actually got to realize that molecules are so impactful, not just within the cell, but they can impact human health, they can impact the whole population, the whole ecosystem. My mind was blown, and I never went back. I was in love, hooked. I was an undergrad, and I thought to myself that the fact that you can spend your entire life chasing what a molecule does, and more interestingly, I think what it did in the past, there was no going back for me. So after that, I started, after I got my PhD, I was actually in medical school, and I was studying proteins, but there was something in me that I think always was very bothered by the way we are studying biochemistry, by the way we are trying to understand the cells. And I felt that we are not really connecting it to the larger picture. That is not necessarily our own, planets. Like I thought that we are not even connecting to the space at the largest scale possible. And around that time, which was about 2009 or 10, I was finishing my PhD. I realized that NASA supported biological studies that seek answers to the questions that I was very deeply interested in. That was very surprising for me because it's NASA, why are they interested in microbes? And that's how I discovered the astrobiology program. And I knocked on the door. Well, I actually did I kicked the door. That's more like it. And I just kicked it, and then NASA welcomed it. They welcomed me. And I've been working with astrobiology programs since then. It's been 10 years now.
And it's wonderful because now your postdoc is a NASA astrobiology-funded postdoc that's now in your lab doing astrobiological research with the tools that your lab have developed. So you're an expert in ancestral gene reconstruction. Another way of saying this is paleogenetics. Can you tell us a little bit about this field in general and how does it apply to astrobiology?
Yeah, so the one thing that was nice about the astrobiology program, and I think that is a NASA spirit too, that they welcomed researchers that challenged their worldview, that challenged the way they view science and the way we study things around us. So I think that's why since the first time I started working at NASA, we clicked visual, at least in terms of vision. And one thing again that was bothering me was that molecules are so impactful. They change our planet as we speak, and they will continue to do so. But we know very little about what they have done in the past. So my postdoctoral studies focused on developing experimental systems that will allow us to study what may have happened in the past that shaped the planet the way we see today. And something that was very special, I think, that happened to me, that doesn't happen to other researchers. There are actually multiple things, but the main one was that I was able to transition from a medical field to a very fundamental, basic area of research that I had no particular training for. But that also brought challenges in the sense that I had to develop everything as we go. So for the past 10 years, slowly and patiently, I've been developing systems that will allow us to rewind the clock, so to speak, and allow us to see how molecules and the organisms that host them behaved in the past. It is quite unusual, I think, because this day and age we are so focused on immediate results and immediate figures and systems that will give products right away. I mean, it's really, it says something about the programs that funded me, that they didn't expect an immediate outcome because they realized some areas of research require years and years of work and development, especially when you are pioneering an entirely new area of study. So not only we are able to predict what the ancestral states of DNA may have been like and what the proteins that we study today and their ancestors may be in the past, we are now able to, we call this resurrection, that we bring them sort of to life, so to speak, to study what they do. It is important to note that we may not be working with the truth, right? So when you're dealing with the past, you may never be able to capture what exactly happened, and that is fine by me, and I think by my group. I'm really lucky. I have really spirited, hardworking team of students working with me right now and postdocs. I think we are aware that it's not the pursuit of the exact absolute truth, but it is getting close to what may have happened, getting a sense of what happened in the past and how the molecules that we see today structured themselves over billions and billions years of time.
So more specifically, you use existing biological machinery of cells and then use computer simulations to deduce what there could have been in the past using the tools of genetics, and then reconstruct those ancient genes, put them in live cells, and see how they evolve back?
Yes. So we do, that's a very good way of putting it, broader sense. We start, you can imagine that we are almost capturing all the languages that all the human populations study today. This is similar to what we do at the DNA level. We try to capture all the DNA that belongs to all the microbial, particularly organisms that exist today, as much as we can, as much as is available at the sequence level. And then we try to walk our way back into the past and try to infer what the ancestral languages were. So in some ways, we are resurrecting ancient languages, just like in Stargates. So we are trying to predict what these organisms spoke in the past and how different it is. And we have a way of doing this in the sense that we are not necessarily going all the way into the past, but we are trying to take snapshots between now and the past. And then once we reconstruct these ancestral genes, and I need to point out that my group is not the only group who are reconstructing ancient genes. It's a well-established field right now, but we are, I think, one of the few, not only these days, apply these ancient genes to understand our planet's early condition. So we focus on DNA and proteins in particular that we think impacted our planet as a whole. So some of maybe any ecologists or even maybe outside the fields, they've been listening to us right now, they may be wondering, wait a minute, you're looking at DNA and planets scale-wise, is this possible? And my answer is yes. Okay, that's why molecules are so cool, guys, like that you can expect a DNA or a protein molecule to impact the planet.
Yeah, and it's extraordinary. And for those of you who are watching, make sure you keep asking questions. Astrobio, if you're watching on Facebook Live, put them in the comments a little bit. Thank you so much for all the questions that are coming in. It's awesome. Really appreciate it. So last month with with Colin, we talked about biogeochemical cycles, how Earth and life have co-evolved together over time, and the air on the planet has changed substantially, and the planet today would look nothing like it did if life had not occurred. And so the tools you're using are able to work out some insights into linking the environment in which those biological products came to be. One of the example is nitrogen fixation. So the process of taking very chemically stable triple bonded nitrogen, breaking it down and bring the nitrogen into the biological cell, because nitrogen is such a fundamental piece of the molecular machinery. And that needed an innovation in enzymes. So enzymes are proteins, which you study. And you've spent a lot of time thinking about how these early molecules that were able to break down this nitrogen evolved over time, and some of the results were quite surprising.
Important to note that entire field of geobiology, also say innovative young age field, and you've done a lot of work on this too, does work on linking what the entire systems, right? So what I think my group brought into this is the integration of artificial systems, artificial biology, actual reconstruction of ancestral genes, and also linking evolutionary biology beyond phylogeny, beyond generating trees. So the fact that we are able to reconstruct not just a version of these proteins, but make a life and screen all the possibilities is, I think, what makes this area, at least for me and my group, I think, exciting, is that we are able to sort of unlock the possibilities of biology. We are not as interested in understanding what happens. That's very exciting, and I want to know. But I also know what could have happened and maybe didn't, and why not. We do, so nitrogen fixation is a system that we study right now, especially my postdoc, Amanda Garcia, also is an astrobiology postdoc. She's done amazing work on this. And we have some cool, new, exciting papers coming out soon. So they are going to be able to post the preprints on this later this summer. So we are tackling the nitrogen fixation and also looking at the metals involved in this and try to understand how elements themselves were selected. Ultimately, our goal is to learn from the past and link what we learned from our planet's past to the observations that we make at the stellar scale. So that is the sort of where the astronomy comes into place. We are looking for life, right? I mean, I've been asking these questions to people. Are we alone in the universe? And I can ask this to people who are listening to me. Anyone out there who thinks we are not alone? Maybe they're typing right now. I don't know. Are we alone in the universe? There's always one or two people, and this is, I can statistically tell you that maybe out of thousands, I get people who think that we are alone. This is it. And I've been thinking a lot about that. And you know, it sounds depressing, but at the same time, it is not. Because if we are in fact alone, then this is the only study biology. So we owe it to our very existence to understand what the hell is going on here. Okay, because this may be the only planet that has biology. You can study geology, you can study physics, history anywhere in the universe, but this may be the only planet where you can study the field of biology, which makes biology an extremely unique science. So what we are trying to, oh, go ahead, sorry.
Oh, I was just going to say, like, so the real value of your science, in addition to looking at modern biology, is that the early Earth is another planet, essentially, for all intent and purpose, is a different planet than modern Earth. But that was very much alive. It was biology that we know was there. And you are looking into ways of how was this biology actually there? And using those metals you were talking about, you can connect the molecular machinery to the environment, and thus some conclusions, which I find fascinating. So it's amazing. Your world is of ancestral gene reconstruction, paleogenetics, is linking with the geochemical space of geobiology. It's a new way of thinking.
Yeah, exactly. So we are not only trying to understand the proteins and how they evolved themselves or how they impacted the environment, but we are also trying to understand how the environment in return shapes proteins and what we can learn about the environment itself by looking at these proteins. I'm not necessarily referring to temperature. I know a lot of studies that try to connect the ancestral proteins sure that's that's not what we're doing right now. We're looking at the cofactors and the different metals that these ancestral organisms, ancestral proteins are binding, and try to make inferences about the environment in which they lived in. We also want to understand what are the other possibilities, and are very important. Is also well known, the area of the exoplanet studies are blooming right now. It is extremely exciting. Our solar system is exciting. When outside of our solar systems is even more exciting. The more we don't know, the more there's work for us. It's great, right? This is a great. It's great to be an astrobiologist because there's a lot of stuff we don't know about, and we are trying to understand by treating our planet as an alien planet itself. We are trying to understand the possibility and the environmental possibilities, in specifically that may give rise to life inside of our solar system.
So your molecular work has also taken you to understand one of the greatest geobiological changes of our planet's history, which has been the rise of oxygen roughly halfway through Earth's history. In particular, you're looking at rubisco, which is the enzyme involved in photosynthesis. And through that, you've been looking at the pigmentation of these bugs. And pigments today, plants are green and the case early on? Is that where your research has been taking you?
Yes. So this is a sort of a joke, but not necessarily a funny one in my group, that we didn't pick anything that's easy. So rubisco is a protein that we study. Same with nitrogenase. It's not very straightforward to study this enzyme biochemically or at the organismic level. We engineer modern bacterial with the ancient rubisco sequences that we inferred to have existed prior to the great oxidation event. So we are talking about 3 million year old proteins that we think has impacted our planet in a very fundamental way. So rubisco is one of them, and we are doing a lot of work with the pigments these days. So not necessarily the pigment that is generated by the cyanobacteria, but actual protein pigments, and how they may have impacted the entire ocean, entire planet in that way, and the color they may have been giving away, say 3 billion years ago. Are they the same? Are they different? The work is getting bigger and bigger, and it is definitely interesting because this is why there's no box. So we're outside the box. Once that the box disappeared. And the comfort zone doesn't exist anymore. So everything we do is opening another area that we don't know about. But luckily, astrobiology is an extremely multidisciplinary, cross-disciplinary science. So we talking to an oceanographer, talking to an astronomer, a planetary scientist is a norm for us. It's we learned the language, so to speak, over these years, and we sort of get together and try to tackle these problems together.
So from this pigmentation of life in the oceans, this is really important for the remote detection of life on potential other worlds. Tell us a little bit about how your science ties to the world of exoplanets that you've been mentioning.
It is not, I should point out that our technology that will allow us to even take a picture of another planet outside of our solar system, we are not there yet, right? So we cannot do that right now. But as a group, I guess we are preparing for the time because that will come. It may take us 2 decades to entirely understand what a planet outside of our solar system, what the actual color might be, but we will get there. So once we are there, I want the research that we produce to be useful. So we are sort of investing in a decade from now where we will be able to detect and observe planets outside of our solar system even better. That being said, we are sort of getting ready for a planet that may be present. But again, the whole premise here is that we are looking for alien life, but our own planet's past is. And I think biologists, we need more biologists to study these problems. We don't, I think, truly appreciate in the education of molecular biology, and I get this a lot because I'm teaching molecular biology to undergrads and grad students. I don't think they appreciate that we are a product of billions of years of evolution at the end. And if we really want to find life outside of our solar system, or out there, regardless of its location, we need to understand two main things. We need to understand how life flourished and occupied our entire planet, but we also need to understand how life originated. I will argue that without solving the origin of life problem, we will never be able to find life. We have to find and make. We need to make life and emerge it ourselves. I think I can say that, and understand the conditions that can harbor life. Okay, so I think for a long time we thought that we will be able to do it. I think about all the, we've been asking like how life started and are we alone in this universe for as long as we gained consciousness as humanity, right? The vital force, Aristotle, Darwin looked at how things changing, how evolution happens. And then comes, we solve the structure of DNA, right? Rosalind Franklin taking a picture, showing us how molecules inside the cell, the very code that makes the entire structure possible, that codes the entire world around us, looks like. Right? We took these pictures, and then comes Miller and Urey, where we are able to spark electric and heat and simulate an environment that may be like ancient planet, and able to make amino acids that are the very founders of the cells, the building blocks. And I think we thought, okay, we got this. We know how change happens. We understand that how complexity works, maybe a little bit, enough to speculate. We can take a picture of a cell, we can drill down, look at DNA, look at organelles, understand how the inside of the cell behaves like. And not only that, but we can make amino acids from scratch. I think scientists thought we got this. Today, we didn't, right? So Miller-Urey experiment was 60s. We haven't made life with over 50 years now. We're still not there. Two, it's important to realize that it is literally hundreds of years of thinking and doing that enabled us to finally come to 2020, where I will argue that we are so close to solving the problem of origin of life. I think we are really close. But I think we were a bit full of ourselves, that we thought this will be easy. And now we know it's far from it.
Yeah, it's not only understanding how those prebiotic molecules came to be and how they transitioned to become biological molecules, but also in what environment did they interact in to allow that communication and sharing of information, which is fascinating. And I wish we had more time to talk about this, but I do want to transition to one of the aspects that makes you stand out as a scientist, is that you are also very passionate about public engagement, about communicating science to the public. After all, you're a co-founder of SegaNet.org, the platform on which you are currently live streaming. So it's fantastic to have you as a guest on the program. And you've been involved for this, and as far as I can know you, at least 10 years.
I am a product of public engagement myself, right? So when the program that I applied to as an undergrad was funded by Harvard Hughes Medical Institute, and it enabled me to spend a summer in a lab and do research. At that time, I was trying to synthesize cofactors in the lab, specifically riboflavin analogs. And I quickly realized that I respect organic chemists, but it's not for me. I can't do it. So, but it was a product of public outreach that gave me the sense of what I can or cannot do, and made me feel like I belong and I have a place in science. And I think that's very sort of realism. And it saddens me that still, 10 years since then, maybe actually more than that, 16 years, we are still hearing from young scholars that they have this inability to imagine themselves as a part of scientific infrastructure. If you are interested in science, then that's very so you into this box of whatever people's perception of science and scientists might be. Doesn't matter. It's their problem, not yours, right? So I am a product of that sort of grassroots outreach. It's Emory, and I think maybe we all have some person that changed the way we think. But Patricia Marsteller, she was the leading person for this group, and even though she was coming from another country, right? I'm Turkish. She's from Georgia, Atlanta. Right, completely different part of the world. I related to her, and it was fascinating to me that there are things that are beyond the languages and cultures and our futures that connect us as humanity. And because I think I was very aware that my presence in science is a product of scientific engagement and outreach, I never even thought about it being a job or a task. It's such a natural thing for me that I feel that it's my job now to give back. So I always did voluntary activities as a graduate school. As a graduate student, I was teaching in Decatur, Georgia. There was a high school there, Decatur High School. So I was partnered with a high school teacher. We taught physics and chemistry for 1 year, which changed a lot of things for me to understand. High school education was mind blown to see kids who live in Atlanta, Georgia, who cannot show me where Canada is on the map. That was a very eye-opening experience for me. And all these things made me realize that I belong here even more, that I have a job and mission, and that is to teach science. And STEGN is a product of that. Funny that we started this, I think, 2011. And it was me being away from my own home country had a lot to do with this, right? I realized that there are all these in Turkey who want to watch astrobiology meetings, but they have no way of doing that. So we started an online group to bring science to everybody. And look at where we are now. It's really amazing.
There's so many more topics. Stop being selfish and open up the floor to the questions, which is one of my favorite part of the show. So let's go ahead and get started. Thank you all so much for submitting your questions. Please keep them coming. Use hashtag AskAstrobio on Twitter, or put them on the chat on segnet.org. If you're watching on Facebook Live, put them down in the comments. We're listening, we're hearing you. Okay, Betül, so are you ready? The first question comes from at Astrosociology, Jim Pass on Twitter. And he asks, what are your thoughts about how social scientists can participate more extensively in astrobiology research, so that the societal implications of astrobiology and SETI can become more mainstream considerations alongside STEM issues?
That's a very good question. So it's outside of my field, so I wouldn't know if the goal of a more social science would be to, in fact, increase the awareness of such studies, or just keep a record of what has been happening. So I'm familiar with studies of Dr. Sophia Roost. She is an anthropology professor, and so she wrote really remarkable articles about how astrobiologists go to field sites and study life. And so there's been some efforts towards sort of documenting what astrobiologists do. That being said, I think we need social scientists to that will be present with us right now. And we know Arsev Aydinoğlu, who is in Turkey right now, has also been sort of studying how astrobiologists communicate and collaborate. But I think the question is a bit broader than this, and I think it's deeper. The question is, whether the social studies can be even a more recognized part of the astrobiology studies. And I will say yes. There are a lot of new astrobiology centers being formed right now. It's an interesting, a very exciting time for astrobiology field as a whole. So if you're interested in this more social sciences part of astrobiology, I recommend that you get in touch with scientists who are leading or who are participating in these NASA nodes, and pitch them your idea. I think we could all use someone who is definitely thinking differently than us, not even as a scientist, but the broadest term as a part of social sciences. So definitely yes.
Thank you, Betül. The next question comes from Hopuri KK, who tweets as @Duduk and he or she asks, who was or what was your biggest inspiration?
Well, it's difficult, again, for me to pin it to a certain person, but I would say that it was more about what happened around me collectively. I grew up in an environment where women weren't given the privilege, and I will argue that it is a privilege, to receive education, and not everybody can receive a really, not even top level, but an education. It's still a problem in our world, unfortunately. So I think seeing what was not given to women drove me to get it on. I was women in my family, they did not have any diploma, so I always joke that I acquired enough diploma for everybody. For all the people that couldn't go to school, I have a diploma for you. So I was just really driven to have my own life, my own path as a woman, and I saw education as the only way to achieve that for myself. Thank you for the question.
Using that article that you were featured in by UN Women, you have another good quote in there that says, your knowledge is your armor. And that ties very well to that answer. Thank you again. Serhat Sefjen on SegaNet asks whether you're taking your experimental evolution research and placing those cells in a paleo-environmental setting.
That's a very good question. So I didn't talk about our experimental evolution studies. The quick answer is yes. We are simulating ancient Earth conditions. Well, once we open the lab again, the goal is to see how the bacteria that we engineer with ancient components respond to the environments that may simulate ancient conditions as well.
Thank you. The next question comes from Marianne Denton, who tweets as @astro_limno, and she asks whether finding a piece of genetic code, RNA or DNA, or a structure like it, be considered on another world, would that be considered as an evidence of life?
Okay, that's a very good question. And so I will respond by quoting Chris Adami, who studies origin of life among many other things. And he talks about finding a screwdriver on Mars, right? So if you find a screwdriver on Mars, will you be convinced that there was life or is life on this planet? So to me, finding DNA or RNA or any molecule that is familiar to us inside a biological cell on planet Earth is like finding a screwdriver on somewhere else. How did it get here? It's so hard to synthesize these molecules, let alone use them as an information system that we see in the cell today. So I will argue that, yes, finding piece of DNA or RNA is definitely going to be, we will be really close to finding life if we find DNA somewhere else, regardless of the context that it is in. If it didn't come from our planet, if it didn't bring it there.
Yeah, it's like finding a hard drive on another world, right? The density of information that's in these molecules are just, it has to be something more than just a natural setting. But who knows, right? I don't think our imagination is broad enough to categorize all the different possibilities. Anyway, so the next question comes from Ulan on Twitter, who tweets as @theysays_Ibrahim. And we were talking about cyanobacteria and how they're parts of plants today. And so he or she asks, are there any plant species that can be grown in space?
So that's a good question too. By space, I wonder if they mean International Space Station, that's also in space, or a different planet. So for the ISS, absolutely, I don't see why not. And but and there are studies towards this, and there are work again towards understanding the conditions that we need to create if we were to grow plants on different planets. There are not just scientific, by the way. I know a lot of artists are also thinking about these things. There's a really fascinating group in United Kingdom that are creating these exhibitions about sort of imagining the future, imagining plants on different planets. So yes, there may be possibilities, but we are not there yet.
Thank you. The next question comes from Burfin Dag on SegaNet, who asks, is it possible to create the genome of the simplest cell that can survive in silico only?
So we can, okay, so there are studies that are done by Craig Venter, for example, synthetic biologists, that tried to create methods to synthesize genomes from, I want to say scratch, but it's not necessarily scratch, right? Because it relied on other organisms. Genome is a template. That being said, there are many facets to this question. Number one is that, what is the simplest organism? Are we talking about the simplest microbe that we have, like a mycoplasma or something like that we have today? Or are we talking about engineering an organism that will have the minimal components that will give rise to a living cell, yet with the minimal components? So if the aspect is number two, we are not there yet. And the problem is, we don't quite understand what exactly needs to be in this bag and by that I mean like the cell itself, that will be minimal but still give rise to life. What is it that we're trying to see? So there's a lot of work towards protocells in that aspect. We are not there yet, right? We didn't make any evolving lifelike protocell system that has information in itself encapsulated. However, there's definitely work towards creating early cells that will have genomes that are not like what we see today. By the way, I don't think it's going to be like what bacteria has. We're talking a lot simpler, maybe less than a couple hundred genes. That is very different. So the quick answer to this question is that we are not there yet, but it's a possibility.
Right, thank you. The next question comes from Twitter. Furikan Kenobi as @SpaceEngineer asks the following question, and he asked it in Turkish. And I would say a big thank you to Professor Arsev Aydinoglu, who has helped us translating live these questions. So if you're watching, Arsev, thank you very much, and thank you. So @SpaceEngineer asks, what steps should a person who wants to become an astrobiologist follow?
Okay, thank you for asking that question, and thank you for Arsev and Sanjoy, because I get that question a lot. So first of all, there are resources that are posted by NASA Astrobiology Institute online that shows different paths that can take you to study astrobiology. But I will give the broadest answer possible, that you don't need to replicate someone else's trajectory to do what you want to do at the end. So that being said, because I studied chemistry and then did biochemistry and then evolutionary biology, you don't need to necessarily follow these steps to study the questions that we do. If you are interested in a basic scientific field, physics, geology, astronomy, chemistry, whatever it might be, do that. And then you will find that there are questions that you can answer that are related to astrobiology, that are embedded in these basic areas of research. I think one aspect that kids are, maybe we're not emphasizing this enough as astrobiologists, is that you need to have a home discipline and excel in that. So whatever you do, if it is biochemistry, then you need to be an excellent biochemist and then build astrobiology on top of that. So you need to have a core area of research. It could be microbiology, it could be geology, it could be paleobiology, whatever it may be. Excel in that, and then apply your technique and your methods to questions related to astrobiology. Because astrobiology is not necessarily a major, right? Right now, nobody offers that. We may do it here in UA, but we're not there yet. So follow the area that you're really passionate about, and then apply what you acquire, your skills and your knowledge towards questions related to astrobiology. Because there's room for everybody in this field. We are not running short of questions. We have so much to figure out altogether, and we will need all of you.
Yeah, being an astrobiologist is really about the questions you ask, not necessarily the science you do. So great answer, Betül. Thank you. The next question comes from Hello Smithers on Twitter, who asks, I am in school right now for environmental science and research with a concentration on genetics. What types of jobs or research would I be able to do when I graduate and want to work for NASA or an organization like it?
I believe this is a student in undergrad level.
I think so.
So I highly recommend that you check out the internship programs that are offered directly by NASA centers, or you could write to professors that are not necessarily working in NASA centers, but are working through NASA-funded programs. And you can directly get in touch with them and ask to work with them, even though it's just over the summer. So I think to get to the job, you need to get to first. I think as a scientist, it will be very useful for us to understand what our interests are. And this is the tricky part about finding your interests. You need to sometimes do things that don't interest you, and that's the only way you will find what you like, through doing things that you don't like. And also finding that the thing that you thought you would like doesn't interest you as much. So I encourage that don't overthink these things. If you like a program, if it sort of sparks something in you, write to the professor, write to the NASA center, seek these internships, and then try to get your foot in the door in one of these labs, and you will walk up from there. But most importantly, also create a mentorship group for you that have your interest, your best interest in heart. These could be people at your level, your peers. These could be people, a mix of both. Build your network and your mentorship network early on, and surround yourself with people who will inspire you, who will share your dreams, and who will join them with you. That's, I think, very important for young scientists. Find a good group that's going to move you.
Yeah, not feeling shy, knocking on doors, and finding a mentor is essential. None of us who are professional astrobiologists did it on our own, right? We all relied on mentors to help us along the way.
I think we think that successful people we're always successful, but that's not very realistic. We are, there's a lot of pain and suffering and heartache and blood and sweat behind any success story that you see. So if you hear answer is a no, don't take it personally. Just keep at it. Don't give up on your dreams. Never.
I couldn't say it in a better way. The next question comes from @ProximaErbo on Twitter as well, who asks, is it possible for a life form with right-handed proteins in different planets? So it has to do with the opposite chirality as we have today on Earth.
Yes, so this is a very interesting question. There are a lot of studies on that. We don't really understand why certain chirality that we see today on Earth is selected. What were the sort of drivers for those sort of selection? I know a very good physicist who did a PhD on this topic, Sarah Walker, and the quick answer is we don't know exactly what the orientation of the molecules that we see today, and whether they need to be in a certain way for them to have the same arrangement that we see on the planet Earth, for them to give rise to life, or at least create the conditions elsewhere in the universe.
Thank you. So many questions. It's fantastic. Thank you so much, everybody, for asking them. The next one comes from @Astrologist, who asks, what subjects should an individual be confident in for studying ancient genes?
Interesting. So I would say, because we start the creation of these ancient genes in the lab through phylogenetics, primarily we want to understand the phylogeny. So the students that join my group, regardless of their background, and we sort of work with these ancient genes in one level, be it engineering or not. But if they're interested in making these things in the lab, I would say evolutionary biology, phylogenetics. And there's an excellent workshop at Woods Hole Marine Biological Labs. Leads it. It's called Molecular Evolution. And I think there is a sister workshop in Greece, in Europe, that also that takes place during the summer, probably not this summer. I encourage you to attend these workshops. They are usually 2 to 3 weeks. It's very intense, but about 10 hours a day you code, and you learn how trees are constructed and all the algorithms behind it. It's very intense, but you get so much out of it in a very short amount of time. So I recommend molecular evolution workshops for anyone interested in reconstructing ancestral genes and phylogeny.
Thank you. So next question by Anurup Mohanty, who asks the same question, but focusing on the technology around reconstructing ancient genes. What toys do you use in the lab to do that?
Okay, so once we infer these ancient sequences, we then we submit these sequences to synthetic biology companies. So if you are in the end of more biotechnology, you could have your own synthetic biology company and work with us and make these genes for us, right? So there are, I think, right now 4 or 5 main companies that we work with in the United States. Once we infer in silico what these sequences are or might be, we then order these genes. So at that point, we move from reconstruction, which is computational, to resurrection, which is more experimental. So we do this on multiple levels because we also engineer genomes, and we use different systems to engineer microbial genomes. If you're going to do biochemistry, we usually order them in, it could be a circular gene for expression, whatnot. So the person who's asking this might be familiar with these applications. If it is for genome engineering, depending on the method that we use, we order linear DNA or again circular in a construct that we are going to be using later on. So so the key is then to know exactly what your application is going to be. And it's also important to note that we optimize these sequences for the organism that we will use these sequences for. So say if you're going to use E. coli, these are the gene sequences are optimized for E. coli, and etc.
Great, thank you. The next question comes from Julia de Marinis on SegaNet, who asks, what was the most challenging part about reconstructing ancestral DNA in E. coli?
That's a good question. The number one thing was that it hasn't been done. It was not done before, so I wasn't sure if it is going to ever work. Whether the system, the genome itself, will communicate with this ancient gene that I'm putting in, so to speak. So that was difficult for me. But I think any engineer knows this, and I did this in biological level, that you will try a million things, and one of them will work, and your job is to find that one thing that works. So I would say there was a more like mental aspect to know, okay, am I just sort of in Turkey we say rowing in your boat on a rock, you know, like am I just imagining this that it will just never work, or did I not try the right method yet? So it took me about 1.5 years to do it, which is long if you think that that's all I did. I did not take any weekends off. I was in the lab the entire time. And again, switching from biochemistry to microbiology, and genomics may have something to do with this because it's a completely new skill. But at the end of the day, I told myself, you know, I had a giant Muhammad Ali poster in the lab that said not only I knocked them out, but I picked around. And I thought, you know, I won't give it up. Like, this is it, so I'll do it. And it worked. And now we're doing it we're engineering these ancient genes in E. coli, in Azotobacter, in cyanobacteria, you know, you name it, we get it to work.
Is that poster in your grad student's lab now?
No, you know, that poster is in a box somewhere because I'm still unpacking.
Tom Caruso on Facebook asks, at last summer's conference at the Earth Life Science Institute at Tokyo, they addressed moving from a disordered system to an ordered functioning state, so moving from geochemistry to biochemistry. Mm-hmm. How does your research connect moving from geochemistry to biochemistry and the topic of molecular signaling between system components responding to a change in the environment?
Okay, that's a very deep question. We don't necessarily refer to these systems as molecular signaling, but I will say that we have this whole emerging area of work in my group, and I work with Zach Adam on this primarily. So all the things I told you about so far focused on life, right? Like life. Even the simplest form is all about life. But then if you think about our planet is about 4.5 billions of years old, and life is, say, the oldest is 3.8, depending on which data you trust, say 3.8, 3.5, 4.1, somewhere around there. So before life happened, there is a state that is completely abiotic. These chemicals are still around us, but we also have life on top of that. So we want to know how that abiotic system transitions into a biological system. And the biological system that we think about is beyond genomes. It's beyond replication. It's beyond basically anything that we can see in the world right now, but it's more about organization and maintenance as a secondary component in this organized system structure. So we published a paper in Complexity about I think last year that we described what are the complexity components that we need to detect and observe in early chemical environments for them to be pinned as biological, right? So that's the real question here. That how do you take a soup that is just chemistry and then infer or predict whether there's going to be life coming out of this? And by life, I don't mean a dinosaur walking out of your test tube. I'm talking about a biological organization. So that's the work we're doing right now. We are looking at the hierarchical structures of early chemical components and how they may be arranged in themselves for them to be pinned as a biological organism at the crux of the very transition. And I don't know who asked this, but send me an email because your question was great.
That was Tom Caruso on Facebook. Thank you, Betül. Great answer to a great question. Next question comes from @CR on Twitter, who asks, perhaps more fundamentally, does an organism need to contain water to be considered alive?
Sine qua non, right? That's what water is, without which nothing. That's we need to constrain our search for life and our definition for life in the universe based on what we see on planet Earth, and the planetary life is depending on water. Is it required? I'm gonna say no, because I want to believe, right? I want to believe in different possibilities, and there are different. There could be different alternatives to this. People have been proposing for my chemistry. I recommend you read, look into those papers. They're very interesting. But so far, life as we know it depends on water, and we need to rely on what we have right now, primarily when we look for life elsewhere in the universe.
Thank you. Andrew Planet on SegaNet asks, is it possible that the exchange of genetic information evolved or happened before asexual reproduction? Or was it the other way around?
So okay, there are many things happening in the early life, right? Even though we started to focus on life happened, there is no life happened, but there's a lot of things happening in between. So it was a very dramatic time, in fact. And when we think about these major transitions, either emergence of, either the evolution of sex, or major transitions in the sense of, multicellularity and all that, I think we sort of don't think about the transitions that predate life. There's many transitions there to exist. That's how I think about it. So we think about last universal common ancestor and what it was capable of, right? It was able to create some sort of ecosystem with its surroundings, but we are more interested in understanding what happened before the last universal common ancestor. So I will say that yes, life has to exchange information, probably in the form of genetics as we see today, but I would argue that probably was much simpler. But that doesn't mean simple. Before Luca and before all the reproduction that we see later on. Answer is yes.
Thank you. So there's a big jump in complexity between reconstructing ancient genes and recreating Jurassic Park, right? But nonetheless, Rohan Shiradonkar, great question on SegaNet, asks, using this tool of paleogenetics, would you be interested in working on a project to bring back the woolly mammoth?
No. No.
Great. Thank you. The next question is by Graham Lau, my co-host here on Ask an Astrobiologist. And he asked the following question. You teach a class of the science of Star Trek. Can you speak to how science fiction gets the science right or wrong with regard to your own research?
Oh, that's okay, Graham. I should have been prepared. I should have seen this coming. So yes, I did teach a molecular biology of Star Trek course, and I will be teaching it again in the spring of 2021. So the reason why I picked Star Trek is that it's not just in science fiction, and Graham knows this as well, that there is a lot of ethical conundrum involved. There's a lot of also accuracy. I think Star Trek, of all the sci-fi TV series or movies that I watched, got science so accurately, despite the fact that it was done decades ago. So I picked Star Trek primarily to expose molecular biology freshman students to origin of life and astrobiology questions. And I think it actually worked out fine because, again, Star Trek, I think, does a really good job of synthesizing a lot of different components. We could talk about what life is, and we were talking about whether data is life or not, whether we have the right to unassemble data, even though it's an AI, right? So this really, I think, was interesting for the students. So I find a lot of similarities, a lot of inspiration. I seek a lot of inspiration from sci-fi when I do my work as well, but I also separate science fiction from science very sharply. I think it's very important that we don't treat a scientific field as a even though maybe here because of the sci-fi inspiration and etc. But I think it's very important that we appreciate and recognize science when we see it, especially for the future of our field. We need to make that distinction very, very clear.
Yeah, I couldn't emphasize more. Science fiction is a great way to get inspired to do science, but it is not science at all. So, Betül, can you believe we've been talking for 1 hour already, and there's just so many questions. So, if it's okay with you, I would like to take 3 more questions, and then we'll wrap things up. Thank you so much. Really appreciate it, and really grateful for your time. I know you're very busy. So, Gaurav Yadav on Facebook asks, can the lipid profile study of extremophiles on Earth be used to identify biosignatures for past life on extraterrestrial planets?
Good question, and I don't study lipids, but I know that my colleague Paula Wielander at Stanford does. I highly recommend. She had an excellent PNAS paper came out a few months ago. They are studying the potential of lipids as biosignatures. And there's a lot of interest in that group, and also the group that she came from at MIT, Roger Summons's group, that studies Martian the potential of finding biosignatures on Mars. So I would like to direct the question towards those researchers that actually study these things.
Yeah. And again, that researcher is Paula Wielander. She's at Stanford University. Next question is by Ahmet Kayiran on Facebook, who asks, does life need to be carbon-based? What other elements could we possibly think of?
So this is interesting. I've never met anyone who did not watch Star Trek who could ask this question, right? So this is why we want sci-fi, because it sparks the curiosity among us. But I also, again, this is in my mind a similar question to whether water is the absolute need. Life, as we know, is only possible based on the elements that we detect and find, and carbon is the primary one. So so far, that's what at least we will be able to find with the methodologies and techniques that we're developing. We are looking for carbon-based life primarily. So don't mark my words. I will be surprised if it is the opposite way.
Yeah, thank you. Great question. Thank you very much. So last question is by Michael Wong on Twitter, who tweets as Mikwai, who asks, you spoke about how a single molecule can have planetary scale impacts. We're seeing something like that today with the virus right now. Is there a way to predict which small pieces of information can or cannot have huge effects on a system like a planet?
So the prediction part is always very fascinating, but it's also really hard. So there are a lot of studies towards prediction in multiple levels, especially antibiotic resistance. Scientists who study the resistance of bugs towards antibiotics have been thinking and building systems that may answer the predictability component of life. And you may be familiar with Stephen Jay Gould's replaying the tape of life experiment, whether we will be ending with the same biota that we see around us today if we were to repeat everything that happened on Earth. So that there's a predictability component there that you may appreciate that it's embedded in there. That being said, I think that there's a lot of computational needs, and we need to first of all understand exactly what sort of circumstances give rise to these sort of invasions. Right? We need to understand the past, so to speak, if we were to understand or come close to predicting what may happen. The challenge with the prediction is that environment changes, right? Environment evolves. So you need to predict the environment, which is really, really hard.
Thank you.
Are there any more questions?
They are, but I want to be respectful of your time. Thank you, Betül. Thank you so much for chatting with us today. It's been an incredible conversation. Our viewers have turned out in large numbers in supporting you and being fascinated by what you research. Thank you so much. So for those of you who are watching, before we call it out, for those of you who are watching, remember that about the astrobiology graphic series that I was talking about at the beginning of the episode. So there is a short premiere happening this Thursday. That's a graphic novel that Dr. Aaron Gronstal is writing. There's a behind-the-scene look at what it takes to make those graphic novels, and Betül features quite a bit in them. So make sure you check that out. I think it's on Thursday. Mike will put up a link for the moment, and don't forget, you can download all these for free. So çok teşekkür ederim, Betül. This is an incredible conversation. I'm really grateful. For those of you who are watching, thank you again. Please share Ask an Astrobiologist to your friends, colleagues, and everybody that follows you online. This show could not be possible without you. So we look forward to welcoming you next month for an episode of Ask an Astrobiologist. And until then, stay curious. Betül, Thank you.